So yeah, very excited to be here to share some of the work we do here at UCLA starting now. So, as Aaron mentioned, I work a lot about, statistical genetics, analyzing those genomic data with the hope that that kind of analysis is going to inform us about the biological insights about the disease, and that can inform about therapeutic intervention. But for the most of the talk today, I'm going to talk about more like a predictive modeling. The therapeutic intervention is not part of this lecture yet. So hopefully by the time I come back next time, I have some students potentially from this audience working on that kind of therapeutic applications so that I can provide some updates. So my PhD training was, Department of Biomedical Informatics, but, mainly thinking about human genetics. So simply said, this discipline is thinking about the relationship between genetic information and phenotypic information. You can, correlate them, adjust covariates, do all kinds of analysis. But there are three main reasons why I think looking at this genetics is a very attractive data modality to think about the biological processes. The first one is the genetics provides some angle for going into causality. If you measure gene expression or metabolites, a protein, etc., sometimes you have challenges to kind of associate whether the kind of underlying biological processes is causing the disease, or what you're observing is actually the consequence of the disease. But for most complex traits, except for the cancer, what you observe in the DNA is actually more likely to be fixed at the time of the birth. So because of that kind of time dependency nature, you have more ability to go into the causal biology of disease you care about. And the second aspect is kind of related to Aaron's very kind interaction. Because we may able to identify the causal biological processes for the disease, that may provide some insights for therapeutic development. The kind of rationale is that if you identify causal risk factor for certain disease, one possibility is to try to kind of reverse the kind of activity of that kind of causal risk factor by developing some fancy compound or immune therapy. And the third angle is thinking about integrating multiple data modalities. Nowadays, there is no shortage of analyzing, collecting data set. You can get hundreds of experimental measurements from expression data, metabolites data, proteomics data. But a lot of them have some genetic basis. So using the genetics as a kind of underlying common language for multimodal integration is actually a very attractive opportunity. And that's why I'm kind of pursuing this research based on human genetics. And this time in the history is great time to study human genetics because there are many large-scale, what we call as biobanks are readily available across the globe. Like here at the UCLA, the medical school people have, established UCLA Atlas cohort. And, across the globe, here in the States, we have all of us research program, medium veterans program, and, other countries as well, like UK Biobank Japan, FinGen. Each have at least hundreds of thousands of individuals. So collectively, you may have access to millions of individuals. Each may carry, genetic information as well as the disease status from electronic health record. Sometimes it has also additional modalities like brain imaging data, blood biochemistry, etc. So using those data resources, people have performed what we call as genome-wide association studies. So I understand this audience is bioinformatics students, but how many of you have heard about what we call as GWAS? 50%? Other 50% are shy? Okay, just briefly. Basically, it's a simple statistical test. Take each genetic variant independently, try to assess whether that genetic variant is associated with a condition you care about or not. So x-axis is a different genomic coordinate. Each dot is a different genetic variant, and the y-axis is a statistical significance indicating that whether the genetic variant is associated with the disease. And because you roughly test 1 million independent genetic variants, genetic loci, the kind of Bonferroni adjusted statistical significance threshold is typically 5 to 10 minus 8. But as you see, this line of the significance threshold is basically bottom of this picture, meaning that there are a lot of statistically significant associations. And the question is now not to identify, how do we identify those associations, but rather, how can we use those genetic variant association to conduct more interesting downstream applications. And one is predictive modeling. Another can be therapeutic intervention. So for the rest of the presentation, I'd like to discuss a technique called polygenic risk score or polygenic score. So I said human genetics is a kind of discipline to investigate a relationship from human genetic data to phenotypic outcomes. And like, you can analyze this relationship in many different ways, but one of the kind of exciting application of this kind of a statistical modeling is predictive modeling. So like a question is, given the personal genetic profile of individual's genome, can you predict, genetic likelihood of developing a certain condition? Or some of the clinically relevant, intermediate traits, such as can you predict high cholesterol, can you predict high blood pressure, etc. Because those factors may inform the causal biology of the disease. And if you're able to stratify those individuals, high-risk individuals from population effectively, you may be able to kind of provide more relevant and timely intervention to those individuals. And typically, people consider a linear model, like, consider weighted sum of the weights of the genetic variant, summed across the relevant genetic variants because of the complexity of the high-dimensional data set. So this technique, genetic score, polygenic risk score, has been first introduced in the literature around 2005. Just one paper searched on PubMed. But last year alone, there are like more than 1,300 publications. Collectively, there are like 7,500 published studies available in PubMed as of yesterday. And this is not just like academic exercise happening in the research laboratory. The clinicians, like medical doctor, publish scientific statement seriously discussing how do we bring this kind of predictive model in clinic. Like this is one example coming from the American Heart Association, talking about when and how should we use this kind of polygenic risk score to identify the kind of patient populations. And this is just a cancer, but there could be potential application for other complex traits once you develop very robust methodologies. For example, application in cancer is another promising area. So why people are excited about this? If you look at the kind of, the stratification of the individuals by genetic risk prediction, you're able to see that this kind of a genetic risk factor can kind of successfully stratify individuals with the high incident of developing a cardiovascular condition. So in this plot, what you see on the x-axis is the age of the participant. So I'm about age of 30, luckily I'm healthy, so there is no heart like irregularity types of condition. But as we age, like 50 years old, 70 years old, some of the individual may develop some like incident of the coronary artery disease. And if you can predict whether I'm likely to suffer from these types of condition early on before you kind of experience the first onset of the symptom, maybe that can be helpful to alter my lifestyle factor, try to do more exercise, eat healthy, sleep well, types of stuff to reduce the risk of developing such condition. So like it's a hard problem, especially for the participant of the younger age group. You don't see the kind of a great differences in the incident rate. But if you're able to predict the kind of a future risk of the disease development very well, that have a great potential to actually improve the kind of life of many people across the globe. So that's a kind of a good side of the story. This sounds like a promising, we have a methodology, let's deploy to the world. But of course, there are some limitations in the current methodologies. One of the major challenge is that the limited transferability of the polygenic score. So, people start to notice that this is more like a AI ethics types of like a question. Like if you train some predictive model from one population, you can assess whether the similar predictive performance can be obtained in other population groups. And because human genetic data are relatively expensive compared to other modalities like paper-based questionnaire, the types of data we have available as of now are mostly coming from European ancestry groups. And if you train the model on, for example, on the European ancestry group, this study by Alicia Martin et al. Back in 6 years ago beautifully showed that the predictive performance doesn't translate well to other population groups. For example, like half of the predictive performance in the ethnic American population or South Asian population. It could be even worse, like a third of the predictive performance for African ancestry population. This is like a really problem, because if you just deploy this methodology without fixing this issue, this may have a kind of a potential to exacerbate the health disparity and have direct impact on and the implications of who's going to benefit from this kind of advancements of scientific research. And as I said, this is mostly driven by the data imbalance available in the field. Most of the data coming from the European ancestry group, despite the kind of the representation of the population across the globe. But of course, the kind of gold standard answer is to kind of promote the more diverse data collection effort across many parts of the world. But at the same time, like we as a computational scientists may able to contribute to this kind of problem by developing new methodologies like statistical modeling or computational tools. So that's the kind of background of the presentation today, and I hope it's clear. If not, I'm happy to take extra time, but how are we doing? Everything good? Everybody's awake? That's good. All right, let's move on. So I have two topics looking at the multi-ancestry model. This is more like a statistical genetics. The another topic is like looking at the genomic annotation coming from the functional genomics, potentially single-cell genomics types of data set. I'll try to finish by some future-oriented discussion. So, as I said, the limited transferability of the polygenic score can be a concern. So obviously, people have recognized this problem like more than 5 years ago. There has been a lot of efforts to address this problem. One of the kind of, the obvious direction is to try to include individuals from multiple genetic ancestry group. If you train a model from European individuals alone, model doesn't perform well. So what if you bring an additional training data set from more diverse parts of the world, and like you can do some simple things like try to train a single ancestry model independently and try to consider linear combination of multiple polygenic predictor to kind of learn the meta predictor. And this has been shown to be an effective strategy to address some of the issue. But looking at the data, this has kind of leave some individuals left untouched from the modeling study. Specifically, admixed individuals who inherit the genetic material from multiple continental ancestry groups are typically not considered in this kind of a multi-ancestry modeling approach because of the multi-continental ancestry nature of the admixed individual. They are typically not fit in the kind of predefined groupings of the different population groups. And those individuals are not analyzed in the polygenic risk score study. So looking at the, for example, European dominant UK biobank data set, up to 10% of the individuals are potentially of the admixed ancestry. And there are some reports saying that 1 in 7 newborn in the US are of admixed individuals. So there is a kind of a critical need to develop a methodology that works for those populations. Scientifically, if you think about the gap of the predictive performance from the European training set and other, for example, Asian or African training populations, predictive performance, those admixed individuals are literally sitting in the middle of those two populations. So if you believe the underlying biology and the genetic factors are shared across many parts of the world, bringing those admixed individuals can provide a unique opportunities to bridge the gap in the current methodologies. So that was the kind of a proposal which I published 2 years ago. We proposed inclusive polygenic score, IPGS, which is polygenic score training using the individuals across the continuum of genetic ancestry, inclusive of admixed individuals. So how do we do this? So the key idea is that we can train this predictive model on the individual level data. So the previous approach tried to group individuals into different predefined ancestry groups, like European group, African group, Asian group, and try to fit some model. But because this leaves out individuals from admixed ancestry, we hypothesize that learning this kind of predictive model directly on the individual level data can address this kind of limitation and actually enable us to bring everybody on the table. The challenge is more like a computational, technical side. Looking at the human genetics data, there are 1 million genetic variants, 300,000 individuals. If you want to load everything into memory, even with the fancy computing resources like Hoffman 2 cluster here, you may have trouble. Like, how can you allocate the kind of a job with a 1 terabyte, 4 terabytes of memory in this kind of a Hoffman 2 cluster? You want to repeat analysis for many different kinds of disease condition. It may not be realistic. So there are going to be some like a methodological innovation that should address this kind of computational challenges. Luckily, through the collaboration with the statistics expert, Professor Trevor Hasty, Dr. Julian Kim, we have previously developed an iterative algorithm that can learn the exact solution of the penalized regression without loading all the data into memory. So I don't have a time to go through all the kind of methodological details, but like, the key idea is that you can, do the iterative procedure. Instead of like trying to fit 1 million genetic variant at once, you can start with like, if you want to pick one genetic variant, what the genetic variant gonna be like? You can fit one model with only one genetic variant. And like once you fit one model, you can consider what should be the next genetic variant that should be considering the kind of predictor model. And you can do the iterative procedure to gradually expand the complexity of the predictor model. And this approach with a theoretical guarantee to conversion exact solution turned out to be very well to address this kind of problem of the computational complexity. So now, with those kind of tools in our hand, we tested this approach in the simulation data set. So like, here we kind of vary the compositions of the training set individuals on the y-axis, and we measure the predictive performance in the synthetic African population and synthetic European population on the x-axis. And there are like two different scenarios of the different level of the polygenicity, how many genetic variants should be involved in the predictive model. And as you can see, inclusion of like minority population, even like 5%, gonna be helpful to improve the predictive accuracy. And motivated by those results, we move on to the application to the real world data set. In our case, we analyze the UK Biobank data set. So the question we are asking is that if you change the composition of the training data set, how would that affect the predictive accuracy of the polygenic score model you develop? The baseline model is trained on the European individuals alone, which I denote as the gray one, white British only model that consider the European individuals alone. The second model is our proposal, like an inclusive polygenic score, which brings everybody from the table because there is no reason to restrict ourselves from working on specific subset of individuals. But of course, if you bring everybody, you have more individuals because you are getting these kind of ancestry groups into the training set. To account for the sample size differences, we have additionally prepared another two training data set kept at the same number of individuals as the baseline model, but has a diverse composition of the genetic ancestry in the training data set. So gray, white British only is the baseline model. Purple inclusive model is our proposal. And the third one and fourth one as a kind of like additional benchmark to assess the kind of influence of the sample size in the predictive accuracy. And like we use like a UK Biobank data set, analyze 60 quantitative traits, adjusted for the standard covariates like age, sex, global genetic ancestry, as well as social determinants of health like deprivation index. And of course, we split the data set into training set, a validation set, and a held out test set to kind of report the predictive performance in the held out test set individuals. So across 60 traits, we first want to look at the kind of predictive performance, average predictive performance gain across 60 traits. So like here are three models that are trained on the same number of 237,000 individuals. And like, the kind of different columns in the plot represent different test set ancestry groups, European ancestry group, non-British white, European, South Asian, African, and other mixed individuals. For the African ancestry individuals, we saw that the diversification of the training set individuals lead to average of 49% improvements in the predictive performance for this African ancestry individuals. This is really exciting. But if you look at the predictive performance for the European population, like there will be a slight droppage of the predictive performance, like negative 2.5%, as expected, because we are training on fewer number of ancestry matched individuals. But what's exciting is that like our proposal, inclusive polygenic score modeling, allow us to include everybody on the table. So if you do include everybody in the equation, like you get to see the improvements for predictive performance for European ancestry as well, like 5%. And for the African ancestry group, we see the average of 60% improvements for the predictive performance. So overall, like our results indicate that the direct inclusion of the admixed individuals improve the predictive performance across 60 traits. And like, this basically the inclusive polygenic training leads to increase in the sample size statistical power. And because of the pooling of the information across multiple genetic ancestry group, that also leads to improvements in the predictive performance across all the population groups. So like so far, I'm just talking about bring more data. That's good. There is not much kind of biological insights. So I'd like to switch a little bit gear about like questioning ourselves. We got some data about what is going on, what we are potentially missing, so that we can learn from the kind of potential failure mode to further improve the modeling effort. So what we presented so far is like, inclusive polygenics for our proposal is try to fit one model for everyone, right? Bring everybody in, fit one model. Hopefully that can work for everyone. So there is a kind of a implicit assumption that the kind of underlying genetic factors, genetic basis of the trait are 100% shared across everyone. But, as in many other things in biology, there could be some exceptions. There could be genetic variants specifically present or enriched in certain regions of the world. And some of the genetic variants may exhibit ancestry-dependent genetic effects. So like by doing the inclusive polygenic score alone, this second component, yellow one, cannot be kind of integrated in the predictor model. So the natural question is, can we develop some methodology that allow us to model the shared component and ancestry-dependent effects together so that like we can learn some additional insights and hopefully that contribute to the improvements in the predictive performance. And if you're running the GWAS analysis for neutrophil counts, one of the white blood cell count measurements, you get to see the kind of GWAS results for a European population on the top, African ancestry individual in the middle. You get to see different pictures. Yes, the statistical power is different, but the African ancestry specific genome-wide association study have a very focal peak. That basically indicates that this kind of analysis may reveal ancestry-dependent association. Indeed, the lead genetic variants reported for this African ancestry GWAS is well-known loci. This particular genetic variant is like a new functional allele for the Duffy receptor gene variant, which is kind of expressed on the surface of the blood cell. And the surface receptor happened to be the entry point for the malaria parasite. So for individual living in the kind of a geological location where the malaria infection is prevalent, having a new allele that shut down the entry point for the malaria parasite is advantageous. Hence, this kind of allele frequency is very, very high in the African population and basically near zero in European population. So like this particular kind of a genetic variant may also have a pleiotropic association to blood cell counts. And those are the kind of biology we may potentially see by looking at the ancestry-specific analysis. And statistically, we are able to kind of test the heterogeneity of the top track and the middle track, running a heterogeneity test. And like, overall, 90 or 95% of the biology are basically shared across everyone. But like you may potentially see small fraction of genetic variants that may exhibit ancestry-dependent effects. So with those kind of notion, there are a subset of genetic variants that may exhibit ancestry dependence effects. Let's think about whether we can bring those genetic variants with the ancestry dependent effects back to the polygenic score modeling. So that from the first part of the presentation, we have a one inclusive polygenic score model that is kind of predictive for all ancestry groups. Additionally, if you care about predicting this kind of blood count cell in the African population, like you may be able to additionally incorporate additional set of genetic variants coming from the heterogeneity test we discussed. This model is feasible because you don't need to consider millions of genetic variants. You can run the heterogeneity test to identify subset of genetic variants, typically a couple hundreds of genetic variants that may exhibit ancestry-dependent genetic effects. So like after feeding this kind of a shared component, you can refit the model by introducing an additional couple hundred variables to recover some of the effects that is not captured in the ancestry shared model. And even further, if you believe the genetic effects of the ancestry-specific variants kind of changing as a continuum of the genetic ancestry. It's kind of weird if you think about biology, right? If you're like an African ancestry group, you have a genetic effect. But if you go one step outside of that kind of ancestry group, like the genetic effect gonna be zero. That's not gonna be biologically likely. So like you can additionally model the interaction between the genetic variant effects with the global genetic ancestry. And like you can do that kind of interaction model as well, because you need to consider a very limited number of genetic variants that may exhibit heterogeneous effects. So bringing this kind of ancestry dependent components into the table, we are able to see the further doubling of the predictive performance for the neutrophil count, showing that this kind of approach is effective. And this particular number, like R square of 0.11, doesn't mean anything. But if you look at the comparison across other population groups, the best predictive performance we obtained for the European ancestry group was a 0.09. With the kind of interaction effects, we obtained the best predictive performance like 0.11 for the African ancestry group, meaning that this kind of more biologically grounded modeling effort sometimes lead to the predictive performance, best predictive performance for African ancestry group, even though only like 1.5% of the training data set in the UK Biobank are coming from this European ancestry group. So like this looks like a hope to me. The kind of course, the training data set is not like diversified yet, but like doing this kind of statistical modeling offers some possibility to improve the predictive accuracy for underrepresented population. And of course, I picked one neutrophil counter example, but in the paper, we presented like 60 traits, and this paper was published a while ago. So to summarize this part of this presentation, so we developed inclusive polygenic score that works directly on the individual level data. Because we work on the individual level data, we are able to bring admixed individuals directly into the training process. If you consider the ancestry shared components alone, we show that like this proposal can improve the predictive accuracy by 60% for African population. And not only just for the African ancestry group, we get to see the improvements for like other population groups as well. And the, as we discussed for the malaria parasite entry point genetic variants, there is a kind of additional procedure we developed to recover some of the genetic variants that exhibit ancestry-dependent genetic effects. And we have those results available in the publicly available browser. Any questions so far? Can we move on? Good. I like people nodding me, so thank you so much for participating. So like to switch a little bit of the gears. So like I talk a lot about using the statistical genetics types of approach, but I recognize that this kind of bioinformatics community, people may work on different kinds of data modalities, right? Like for the first part of the talk, I talk about leveraging a statistical pattern from ancestry diverse individuals. All you care about is like a SNP genetic information, a phenotypic information. But like there are a lot of data modalities measured in between, like chromatin accessibility or like histone modification from different kinds of biosamples, tissue, cell types, and disease context as well. So in principle, those kind of additional data modality should capture some aspects of the biological processes, and that should, in principle, inform us about the predictive modeling effort. But there can be some practical challenges. Looking at one of the largest effort of the consortia effort, such as the ENCODE consortia phase 4, there are like different kinds of annotations coming from different biosamples, different kind of resolutions of annotation, sometimes variant level characterization, other times like region level, 200 base pair level annotations, and different kinds of experimental or computational modalities. And it's not practically clear how to select which one is the most relevant modality for statistical genetics modeling, and how do we bring those kind of modalities into predictive modeling, like the one I presented like 5 minutes ago. So to address this kind of challenge, how to pick the most relevant data set, we team up with the researchers from MSKCC and use the kind of a transfer learning approach to inform the polygenic score modeling using this kind of multiple data modalities. So as I said, the question was how to pick the most relevant modalities from different kinds of annotation, for example, from the ENCODE consortia. So our proposal is to try to learn the optimal combination using a pre-training strategy. So on the left, we may get the fine mapped genetic variants from large-scale consortia, like Millions Veterans Program, that can identify likely to be causal genetic variants across hundreds of traits. You get to see the likely to be causal fine mapped causal variants, that's a positive label, or non-causal variant, looking at the kind of new result genetic variant with the causal likelihood of less than 1%, for example. That can be a negative label. So using this kind of positive and negative as a label for the pre-training, you can learn the optimal combinations of the epigenomic annotation that is informative of the kind of functional genomic profile that is coordinated with the causal variants in the statistical genetic settings. As aggregate, we get to see like what we call a CVTF score, consensus variant to function score, representing the kind of aggregated support of the more likely to be causal variant for each genetic variant. And like we took this kind of a CVTF score to transfer learning. Like again, we can fit the polygenic score modeling, for example, in the UK Biobank. And when feeding it, like we can adjust the kind of prior likelihood of including genetic variants proportionate to the CVTF score we learned from the pre-training stage. And like by doing this kind of approach, we are hoping that we hope that we are able to improve the predictive performance of the genetic predictor model, because our hope is that we are able to prioritize more likely to be causal variant. And because I'm talking here, this approach actually works. And this kind of approach integrates three orthogonal methodologies. Like we try to take advantage of the genomic annotation as well as the biological knowledge. And also like a second, we like to prioritize likely causal variants via statistical fine mapping. And the third, we are trying to do the multi-ancestry polygenic score training as we saw in the previous work. And this was led by Lucy Tian, who was a master student back in MIT Harvard, but now she's a second-year PhD student at Cornell University. So with this kind of setup, we are able to integrate different kinds of data set, like ENCODE consortia data set, Million Veterans Program, fine mapped variants into the predictive modeling. And like, once you have the pipeline sitting up, like you can ask a lot of questions. Like there are several choices, study design questions you can ask. For example, if you want to train this kind of a transfer learning framework, would you like to focus on the all kinds of annotation available in the ENCODE dataset, like coming from all kinds of different cell types, cell line, tissue, biological samples? Or would you like to focus on specific annotation coming from specific biology of interest? Similarly, you can look at the different kind of ancestry groups used for statistical fine mapping in the Million Veterans Program. Would you like to learn the kind of combinatorial feature of the functional genomic annotation from all the causal variants from the MVP cohort, or would you like to focus on fine mapped variants coming from European ancestry alone or African ancestry alone? And we can compare against existing polygenic score models. And like going back to the original question, what kind of functional genomic annotations would be most informative to improve the predictive accuracy of the model? So Lucy investigated these kind of questions one by one. The first one is like looking at the effects of the genetic ancestry in the fine mapping stage. So like, we assess the predictive performance for like 4 traits in the UK Biobank population. And like we ask whether we should use the fine mapped variants from the African ancestry, which is ancestry matched, or European ancestry group, which is ancestry unmatched. And because of the kind of data imbalance, if you use the fine mapped variants from the European ancestry group, you have more variants available. Like you have about like 5.7 times more fine mapped variants available, because you have like 4x more individuals in the fine mapping analysis in the Millions Veterans Program. But despite this kind of a 4 to 5-fold differences in the availability of the data set, Lucy demonstrated that the working on the ancestry matched genetic variant set is most promising to improve the predictive accuracy. Similarly, you can look at the different kinds of annotations from the ENCODE consortia. You can grab histone modification and chromatin accessibility from all kinds of biosamples, or you can focus on the kind of annotation coming from specific biological samples. For example, for predicting a lymphocyte count, which is a count of the blood cell, you may want to use the, the histomodification as well as the computational prediction from the blood samples instead of bringing everything together. And like, similarly, if you're analyzing the kidney function, you may want to use the kidney-specific annotation by removing irrelevant data points. And similarly, like, if you focus on the tissue-specific annotation, you have typically fewer number of data points available in the pre-training. Like if you grab everything in a consortia, you may have access to 400 data sets. But if you're looking at the kidney-specific annotation, you only have like 11 data sets, like 40-fold difference. But still, we found that looking at the tissue-matched annotation is most informative in improving the predictive accuracy. So like combined together, there will be like 6-fold differences in the ancestry. There will be like a 20-fold differences in the tissue-specific annotation. Combined together, there will be more than 100-fold differences. But like we demonstrated, looking at the biologically aligned integration of the ancestry-specific and tissue-specific integration is the most beneficial strategy to improve the predictive accuracy. So like this basically indicates that this kind of framework offers the pragmatic solution to kind of take advantage of the available resource as of today, while waiting for the data generation from more diverse biological contexts. And like in terms of benchmarking, there are like different methods that consider different kinds of ideas. Like we consider all of the 3 approaches, looking at the genomic annotations, causal variants, as well as a multi-ancestry training. There are like a few methods that consider some flavors of those ideas, but with the comparison with other methods, we show that like our methods rank the best or at least comparable within the error bar margin, highlighting the kind of integration of those 3 major ideas are basically effective. And lastly, because our models are like trying to learn the optimal combinations of the epigenomic annotations, we set to ask whether we can find one single modality that is most informative for improving the predictive accuracy of the polygenic score. Like we can look at the feature importance score types of analysis, comparing different kinds of annotations on the x-axis and the different kinds of genetic variants on the y-axis. So if you were to see one single annotation modality informative to improve the predictive accuracy, we should see one single vertical line representing that this feature is most informative to select all the genetic variants. But what we see here is like somewhat blurry heterogeneous profile, indicating that like picking one modality from the consortia effort is not sufficient. Using this kind of optimal combination strategy as the one we proposed here is the kind of necessary to bring the information for prioritizing more likely to be causal variants in the predictive modeling. To summarize this part of the presentation, so we presented the PRISM, the transfer learning approach to improve the polygenic score transferability by systematic integration of diverse genomic annotation from the consortia types of effort. Our application focused on the ENCODE version 4, largest integration today, focused on like 414 data sets, as well as the largest today 5-mapped variant set from the Million Veterans Program, and demonstrated the predictive performance improvements in the UK biobank traits. After developing this kind of pipeline, we demonstrated the biologically aligned integration, ancestry-specific, tissue-specific integration is more effective despite the 100-fold differences in the availability of the annotation data set. And we saw that the average of 13% improvements for African population group over what we presented in the inclusive polygenic score model. And because of the modular nature of the design, this approach allows the biological interpretation, allowing us to ask what kind of genomic annotation is actually contributing to the predictive modeling. So hopefully those are the kind of some flavor to illustrate not only just looking at the statistical genetics, but like getting some like a rounded training across different kind of data modalities can expand the toolkit you can use in the research settings. And I talk a lot about looking at the linear model, but there are some works. We look at the non-additive genetic effects, looking at the additive effects, recessive effects, genetic dominance effects that can be nicely integrated into the polygenic modeling in the statistical boosting techniques. And we are going into nonlinear genetic effects by asking like gene by sex interaction effects incorporated into the predictive modeling. There are like some exciting results showing that the considering gene by sex interaction actually further improve the predictive performance for the obesity-related traits for African ancestry groups. With that, I like to kind of discuss what's the kind of like future directions. And I talk a lot about polygenic score. It's to summarize, there was a kind of statistical approach to aggregate genetic effects across many different genetic variants. And I introduced several of the ongoing and the future directions, thinking about the multi-ancestry modeling, as well as I didn't talk much about it, but like there will be some exciting effort looking at the multi-trait modeling, and like integrating like biological annotations, genomic annotation, as more frankly, domain knowledge integration into the predictive modeling. Nowadays, potentially with the help of the large language model, can be a really exciting direction to align whatever we develop with a statistical modeling, actually grounded on the biological common sense. And there are some directions we can go looking at the context-dependent nonlinear effects, looking at the interaction of the genetic effects with the genetic ancestry, as we discussed in the inclusive polygenic score, or gene by sex interaction, or G by exercise, G by environment interaction. Those kind of an integrative machine learning model can provide a unique opportunity to kind of incorporate time-varying covariates as we kind of progress through the aging processes. So like, there are a lot of like open questions and potentially opportunities for you basically, thinking about, how do we kind of consider the heterogeneity in the disease? So I talk a lot about like predicting disease or not, but like, disease is not a monolithic entity. Some people have a faster progression of the disease, other people don't. Some people respond to the treatment, other people don't. Like, can you go beyond the simple case control comparison to think about, can we kind of inform some predictive insights for heterogeneity in the disease? And like for this kind of like a more nuanced inference, it will be more informative to think about the accurate and calibrated and explainable machine learning and AI modeling for polygenic risk modeling. And like this contextualized prediction, G by context, environment, different kind of like demography is more relevant. Like if you're living in the LA region, like is the genetic risk can be modulated by exposure to air pollution, for example. Those gonna be the types of questions which we frankly don't know much about. And like, this is all about predictive modeling, but like, can we align more to the causality? Genetics provide a unique angle for causality. So like if you have a prediction, can you integrate the kind of causality into this kind of predictive modeling so that like whatever like a stratification you construct actually inform the intervention in the patient care setting? And I talk a lot about the methodology, but of course, disease-specific applications for application to specific biological processes like aging will be a potential interest of the kind of research in the future. So with that, like, this is my new lab, like, trying to address a lot of related questions in the context of disease heterogeneity. And like, we are lucky to have exciting first member, like 3 talented students, 1 each from PhD, master, and undergrad. And frankly, I'm looking for more members, like any potential rotation students or any of your friends looking for postdoc, PhD, etc. I would love to chat with you after this presentation. I'd like to thank my current member and the past and present mentor mentees and our collaborators. And yeah, happy to take any question. Thank you so much.